The building blocks of DNA are the fundamentals of how a cell works. In the normal cell, the DNA makes everything happen that should happen. In the cell that has cancer properties, the DNA makes the odd things about a cancer cell happen. So if we can identify every single DNA base and figure out where each one is and where it should be, and what's changed about that in cancer, then we can figure out what's gone wrong at the functional level.
We're made up of billions of cells, trillions of cells, and each one of those cells has, in principle, the same genetic code. It's the code that we were created with as a single fertilized egg, and that determines who we are. Every single cell carries that genetic information with it. But if you look at our bodies, we are composed of very different cell types. And if you look at the skin versus, say, a liver cell, they have exactly the same DNA sequence, or for the most part. There may be an occasional mutation that's irrelevant, but for the most part, they're identical genetically, but yet they have very different phenotypes. They have very different appearance. So the cell in a skin cell looks very different from a liver cell. And so the question is, how can exactly the same DNA code give very different appearances, function, behavior of cells?
Most of human DNA is constant between individuals. So if I look at my DNA sequence and yours, it's virtually identical. There are certain sites at which it will differ. A lot of these sites have absolutely no effect at all. They just happened by random chance. A lot of the times, however, a DNA site, a given position, can be changed in some way. So an A can change to a G, or a whole chunk of A, G, C's, and T's could be deleted or missing. In cancer, we look for particular cases where this kind of change occurs in an important gene or an important pathway that might indicate that changing that gene is what led to that cell becoming a cancer cell.
We know that cancer is a multi-step process, and cancers can be broken in very different points in their sort of in the normal mechanism of how cells live and how cells function. DNA tells us a lot about where the broken parts are, and that's critical for targeting our fixes. For example, if your car's making a rumbling in the front, you need to know if it's in the engine or in the transmission. And that's sort of what we don't know currently. Is we know that these cells are broken in some way, but we're not always able to tell where the break is so that we can target it with a therapy.